This is passage 2 of the AAMC section bank volume 2, and this is for the biochem section. The influenza A virus, IAV, is a negative sense single-strand RNA virus. So this is our virus information, so it's a good thing to highlight. IAVs are divided into subtypes based on the presence of different combinations of their surface proteins. Hemagglutinin, or HA, and then neuraminidase, NA. Each IAV subtype contains one HA subtype and one NA subtype. Okay, and so the researchers, they said they identified two types. There's the 18 one and there's 11 one subtype, and this was done through Western blotting. Okay, and so I think it's a good thing to know to highlight because it's a content review term. So when I'm going through these passages, it's always good to like highlight familiar key terms that you know from your content. And so then they're saying this virus affects mainly the lining of the upper respiratory tract. And then they said, however, different host species are distinct into their susceptibility to varying IEV subtypes. For example, IAV subtypes containing HA1 can affect birds and humans, whereas IAV subtypes containing H11 primarily affect birds but not humans. Okay, and so they just tell us the differentiation, like what do these different subtypes do? And so they said to infect a cell, HA attaches to salicylic acid on the cell surface, after which the IAV particle is internalized via vesicle formation. Okay, another key term right here. I'm thinking right away endocytosis or exocytosis. And then salicylic acid is a monosaccharide, okay, another key term, links to the terminal galactose and glycogen chain via this through alpha-2,3 or alpha-2,6 linkage, and then birds predominantly express a alpha-2,3 linked to salicylic acid, whereas humans predominantly express alpha-2,6 linked to salicylic acid. And so they said right here, they're talking about carbohydrates, another key term right here from our content. And then so they said once inside the host cell, these new proteins are produced and synthesized, and the virus genome material is replicated. And then microtubules transport newly synthesized proteins and synthesized IEV proteins and genome to the cell membrane, where the virons are assembled and bud from the cell. Finally, Na cleaves salicylic acid from the components of the cell membrane, thus allowing the release of newly assembled IEV virons. Okay. So they conducted, now they're saying there's several strategies to treat and prevent IEV infection. These include this, which directly prevents the release of newly assembled IEV virons from host cells, and then influenza vaccines are also available, including subunit vaccines, which preventatively expose individuals to HA and NA proteins. Question 1 of passage 2. Which transport mechanism does IEV use to enter a host cell? So recall that from like our second paragraph where we highlighted internalized via vesicle formation. So that tells me right off the bat it's could be like something with the endocytosis. So let's look at our answer choices. So I can see right away that endocytosis is already our answer, so I'm going to select that. But for the sake of completion, I'm going to explain what do these answer choices mean. Okay, and so A is wrong because exocytosis is the opposite of endocytosis. It's when like the molecules they form vesicles and then get released outside the cell. And so in this case, our virus is trying to get inside the cell using vesicles from the cell membrane of the host cell. So it cannot be A because it's trying to go inside, not outside. And then C, simple diffusion. So if you recall from your content review, like this is our, let's say, our cell membrane, right? And then you have something like O2, a gas, trying to simply diffuse through. These can go through easily because of their chemical structure, so it allows them to go through easily instead of going through a like a ion channel. And so, but the thing is, however, viruses, they're much larger than oxygen, and obviously they can't go through simple diffusion, so C is not the correct answer. And then facilitated diffusion, it's basically like simple diffusion, but it's with a ion, like a channel, a carrier channel, you can call this. So something that will go through a carrier channel is something like maybe like sodium or potassium. These are like more likely candidates to like go through a channel rather than simply diffuse. And so, but however, like the virus is much larger than like sodium and oxygen combined, and so it's not gonna be able to go through like facile diffusion because it's too big, and so it has to go through something much larger, in which case endocytosis, which is reserved for like larger molecules. And so that's why B is the correct answer. And then that's question number 1. Question number 2 of passage 2. Which mechanism is used for the synthesis of IEV proteins? And so recall back from the passage that they said, when they first talked about the virus, they said it's a negative sense, single-stranded RNA virus. And so this should be enough information when we're going to solve the problem. So let's go through the answer choices one by one. A. Direct translation of the viral genome. And so recall that this is something of a behavior of a positive sense RNA virus. So if I were to like bring a drawing up right here, so to compare the two, a positive sense virus, what it does is when it first enters the cell, it uses its RNA and then it gets directly translated to protein. Because reason is because the type of RNA it has, it's something that the ribosome can recognize. And so it's able to use its machinery and go directly into translation. But if we look at the difference between, now we're looking at negative sense now, negative sense virus. These guys, so they come in as negative RNA, and then they have to go through this thing called transcription. So when they transcribe, they get converted into a positive sense RNA, and then only then that RNA can be used to be converted into proteins to build the virus. And so then it has to be transcribed first for that to happen. And so let's go back to your answer choice. So this is basically what we just said, positive sense, not negative sense. So this is wrong. And then let's look back at our let's go to our other choice right here. Translation of transcript. Transcripts formed using RNA-dependent RNA polymerase. And so what are they saying? It's basically what they're saying is that if you look back, they said RNA-dependent RNA polymerase. So they're basically just changing the type of RNA that's used. So it's something that lines up with the negative sense. So it goes through negative RNA, comes as negative RNA, and then it used the polymerase, and then it converted to something similar to that the host cell can recognize. And in this case, positive RNA, or you can call it mRNA. And so then this directly gets recognized by the cell, and then boom, it just makes proteins. And so this definitely lines up with the negative sense type of virus. So B is a good candidate so far, so I'm going to hold on to B. And now if you look at C, transcription of DNA formed using RNA-dependent DNA polymerase. So this is something of a behavior of a retrovirus. And so what does a retrovirus do? It takes its RNA and then it uses reverse transcriptase to convert to DNA. And that DNA then gets integrated into the host cell genome, and then it converts into, and then uses the translation. When you have like a negative sense or single-stranded RNA virus, these types of like viruses, they just go like, they don't even like integrate themselves into the genome of the host. They just convert their negative sense into a positive sense, and then they directly translate using ribosomes. They do not get integrated into the genome. And so C is not a good answer so far because of that. Now let's look at answer choice D. Direct translation of our genome using DNA-dependent RNA polymerase. And so this is what it's talking about as what's called a DNA virus. So if I put like a diagram right here, so a DNA virus, it converts its RNA into DNA. It's DNA to RNA, my fault. It's the other way around. So like the virus, it comes in as, and let me show it like a drawing right here. Once more, the virus comes in as the form of DNA, and then it gets translated, transcribed into RNA, and that RNA is used to make proteins, etc. Right? So then, but this is, we're dealing with the RNA virus though, not a DNA virus, and so these are the incorrect answer. Just leave in B, and then B is our final answer, and that is correct. And then that is question number 2. Question number 3 of passage 2. Which response most likely occurs in an individual to whom a subunit vaccine has been administered? So if we recall from our last paragraph from the passage, they discussed like treatment strategies, and so this include using a subunit vaccine. So what is a subunit vaccine? So just to kind of like a brief overview, it's a type of vaccine that like, let's say you take a piece of a virus, right? Let's draw a piece of our virus right here, and then you put it in a form of a vaccine. So I'm going to draw a needle to represent that. Try my best needle. And so this kind of represents the vaccine. So if you inject in a person, they get exposed to a piece of that virus. And so when they're exposed to that piece of virus, the human, what it does, it produces the necessary components to defend it off. So this includes producing antibodies, um, creating antigens. And so in the case scenario when the body like encounters a real virus or like a real infection in the near future, it has these things prepared, and so it's able to quickly respond to the threat and able to neutralize it before the person gets sick. And so that's kind of a brief rundown of how vaccines work. And so let's go through our answer choices. And so they said, let's look at choice A: production of antigens by T cells. So recall for your content review, T cells are those guys that like they respond to antigens. And so they release, once they respond to an antigen, they release these things called cytokines. So these cytokines, they destroy the threat. And so, but T cells don't produce antigens. That's the job of the macrophages. They consume the threat, and then they display their antigens outside of their cell membrane. And so this is something the T cell responds to rather than produce. So A is not a good answer. And then B, production of antibodies by T cells. And then recall for your content again, production of antibodies is the job of beta cells, not the T cells. And B is wrong as well. Now, let's look at C and D. And so I kind of want to do a little explanation here. So they're saying in the problem that plasma cells. So what the heck are plasma cells? And so I want to show the difference between plasma cells and beta cells, or B cells. So plasma cells, they're the cells that produce antibodies. They're the ones that already have recognized a antigen from the macrophage, and then they produce these antibodies. And so they will continuously produce them. And now a B cell, it's a cell that that's not producing any antibodies. It's kind of in its neutral state, and so it will not produce any antibodies ongoing for the specific antigen threat, right? So right now, it doesn't have any producing antibodies, and so that's just kind of something to know when you're going to MCAT, is just to know that plasma cells and beta and B cells are the same thing. It's just that plasma cells are the ones that produce antibodies, while B cells really don't, because they don't really have the machinery yet to produce it, if that makes sense. So let's go to our answer choices one more time. So this says production of antigens. Again, they're producing antibodies and antigens. That's the job of the macrophage. So that's on C. And then if you go to answer choice D, production of antibodies, plasma cells, that lines up from our way of thinking, and so D is the correct answer. And that is question number 3. Question number 4, passage 2. Which step is not involved in the technique mentioned in the passage that researchers use to identify IAV antigens? And so they're asking not. So it's always like good to like highlight what's not involved. And so recall that from our first paragraph, they said they separated the proteins by Western blotting. And so they kind of just like differentiated by molecular weight. And so I want you guys show you guys something that, because there's also these other types of blots, and so I want to show you mnemonic to help you remember these types of blots. And so it's always good to know on the back of your pocket, because you'll be tested in all types of blottings on the MCAT. So remember this mnemonic: snow, drop. And so the S stands for south blotting, and then N stands for north blotting. You can omit the O, and then there's the W, which is western blotting. That's for all of these, so you guys can know. And then this is for DNA, D is for DNA, R is for RNA, and then O is omitted, and then P is for proteins. And so if you go these threes one by one, south goes with DNA, north goes with RNA, and then western blotting goes with proteins. And so it's a good way to remember, like, which blotting does what. South does DNA, north does RNA, and then western blotting, in which in this case is doing proteins. And so it separates proteins by size. And so let's go through our answer choices and like identify which is not involved in the process. So I have as we go along, I have this diagram right here, which kind of explains well how the process works. So just to give you a quick rundown, you basically take your sample, your protein sample, and then you run this thing through gel electrophoresis, and then you run a current, and so then it separates the proteins based on like high molecular weight and then low molecular weight. Obviously, the low ones go straight through, and then the high ones stay back up at the beginning of the cathode. And then once you're done, you kind of go through this thing called membrane transfer. You kind of plot it on a a plot, and then you use like antibodies to kind of like differentiate like which proteins like are being shown. And so let's go through our answer choices. So what is not involved? DNA sequencing. Well, we said that proteins, they're the ones that are being used here. That they're the ones that are being separated. So there's no DNA sequencing happening. That's more from like when you see like if you go back to our mnemonic, that's more for like southern blotting, not for western blotting. So A is something that it's a good candidate, and so we'll keep that for now. And then membrane transfer, that's something we just talked about just now because once you have the protein go through, it goes through after gel electrophoresis, it goes through membrane transfer. So B is a still correct answer. Gel electrophoresis, we just discussed just now, it goes through a current, gets separated, so that's part of the process as well. And then D, antibody incubation, and so that is another process too because at the end of the blotting, you have your proteins like identified through antibodies, and then there's a period where you have to like incubate them before you can actually see what the proteins are, and so that's a part of the process as well. And then A is our correct answer, and then that is question number 4. Question number 5 of passage 2. When acting as a HA receptor, which type of bond links salicylic acid to its associated oligomer? And so let's look through our second paragraph where they first talk about acting as a HA receptor. And so to infect a host cell, HA attaches to the salicylic acid on its cell surface, and after which the virus particle is internalized via vesicle formation, and then it's saying that the salicylic acid is a monosaccharide that links to the galactose. And so this whole process right here is telling me something to do with linking through carbohydrates. And so that's something I'll go in mind when I'm going through answer choices. So let's look at A, disulfide. Disulfide is basically when you have two cysteine like amino acids being bonded together. And so, but this however is talking about carbohydrates. They're showing like a glycan chain and stuff. And so we cannot say it's A because this is talking about proteins, this is talking about carbohydrates. B, glycosidic. This is a good answer because this is basically when you have two uh, glycogens, right, and they make a bond with each other. And so this is kind of similar what they're talking about between linkages, and so this lines up the most, and so we'll keep B for now. And then let's look at C, the peptide. And so peptide bonds, kind of similar to answer choice A. It's when you have two different amino acids being linked to each other. But this is talking carbohydrates again, and this is proteins. And so C is not the correct answer. And then yeah, D, phosphodiester. Um, this is something you typically see in like ATP, like you have your your three phosphates being bonded together in ATP, but this is talking about carbohydrate chains, and so this has nothing to do with phosphodiester, and so D is not our correct answer, and this just leaves B, and so that is question number 5. Question number 6 of passage 2. Which tissue is most likely infected by IAV? So if you recall from the first paragraph, they said that IAV primarily infects the lining of the upper respiratory tract. And so this respiratory tract, it's kind of known to catch like mucus, any foreign particles, and kind of like stick to like the brushes of the cell. So let's go through our answer choices one by one. So A, aerial tissue. So that's like in your tissue. This is mostly common in your skin. It basically connects the skin of your skin to your muscles and kind of keeps them together. Has nothing. It's not really like existent inside the upper respiratory tract. So A is not a good answer so far. So B, adipose tissue is basically mean like saying fat cells. Like that's where your like fat is being stored and by your fat cells, and they comprise of tissue. And so this is in your upper respiratory tract. There's no fat lining in your upper respiratory tract. And so B cannot be the correct answer. And then transitional epithelium. So I have a good like diagram right here. So this is kind of something for the MCAT to be familiar with all types of like epithelial tissues. There's a squamous, cuboidal, etc. So if you look at transitional right here, it's like comprised of different types of like cell shapes, like simple or squamous. But one thing you want to notice is between the difference between pseudostratified and transitional is that pseudostratified, it has these brushes right here. These brushes are very common inside the upper respiratory tract because, again, like I said, it like holds mucus, and this is where it catches foreign particles and kind of like make sure it doesn't reach our our lungs at all. And so this is something that you expect to see in your lungs and or your respiratory tract. And so I would say D is a good answer. Transitional is wrong because it doesn't have these brushes, because if it if maybe they do exist, some of them inside the upper respiratory tract, but majority of the time it's gonna be these brushes, because without these brushes, then we'll be like sick a lot, and then we'll have a hard time like kind of breathing, because you don't have a mucus line, and so D makes the most sense because of the brushes, and then C doesn't because it doesn't have those brushes, and so D is the correct answer, and that is question number 6. Question number 7 in passage 2, and then this is the last question. Based on the information in the passage, what is the most likely conclusion regarding the interaction between HA and salicylic acid? So going through these, they talk about HA1, HA11. I'm just kind of skimming through my answer choices a little bit before I go back to the passage. And so they're talking about something that looks like to me like something about carbohydrates or like some linkages going on. Okay, so let's read our first sentence of let's read the last sentence of the last paragraph, first paragraph, and then kind of going on through this middle paragraph right here. And so then they said the subtype containing HA1 can infect birds and humans, where HA11 primarily infects birds but not humans. So they're kind of like showing us a differentiation between these two. HA1 does birds and humans, while H11 does primarily birds. Okay, now if we continue down, they said that for birds they express mostly a 2-3 linkage, and then for humans right here, they said that they express a 2-6 link to salicylic acid linkage. So this is for humans. Okay. Now let's go back to our question right here. And so let's look at answer choice 1. H1A rarely binds 2,3 linked salicylic acid. So basically what they're saying is the 2,3 is it belongs to birds. So they're saying that this subtype rarely binds to birds. So I would say this is wrong otherwise, because remember, if you go back to the first paragraph, they said it can affect birds and humans. And so this cannot be correct, because rarely is saying that it almost never binds. And so in rare cases it does. But HA1 infects birds and humans, so it cannot be A, because it does bind to bird linkages. And so now let's look at B. Same thing now, but they're saying that this is for the humans now. The humans, they have a alpha 2-6 linkage, and so remember they bind both birds and humans, and so saying rarely means that like almost never, and so B has to be wrong also. And now if you look C and D, C is saying that HA11 preferentially it binds to 2,3 alpha 2,3 linked salicylic acid over 2,6. So basically, if you recall again, like HA11, we highlight where it says that it primarily infects birds, and so our focus is that it wants to infect birds over but not humans. And so 2,3 is the birds and 2,6 is the humans. So you're saying that it preferentially binds birds over humans. I like the answer. It makes a lot of sense because 2,3 is for birds and 2,6 is for humans. And now I like C. We'll keep a hold on to that for now. And let's look at D. It's the same thing as C, but it's kind of flipped the other way around. It's basically saying that this subtype virus likes to bind to humans over the birds. And so they said that infects birds but not humans, and so D is not a good answer, and so C is the correct answer. And then that was the final question for passage 2.